I'm the Collection Manager for Malacology at the Australian Museum, so that's all the shells, mussels, bivalves and cephalopods, squid, cuttlefish and octopus come under my domain. The mollusk collection is so large that we have a separate section, even though we do look after marine invertebrates. We have our own section at the museum. We've got about 11 million mollusk specimens at the Australian Museum, so it's something that keeps you up late at night worrying about that. So what I just wanted, some of this might sort of be repeating what Letitia said, but I just wanted to outline what we actually do in the field and how do we use museum collections afterwards. So next one. So what we do is for large invertebrates, they can be hand collected, like this octopus, which wasn't under the wharf. That's actually from French Polynesia. But what we need to do to find a lot of cryptic and small creatures is to collect the substrate that they live in. And as Letitia said, we bring that back to the lab, pick through it, sort through it. So, for example, this little nudibranch lives in Calopa, but you, just by doing a visual swim over it, you're not likely to see it unless you look at it under a microscope. Next one. So sorting. This is why we've got to rush off, unfortunately, tonight, because we did a lot of collecting today, so we're probably going to be up till quite late tonight doing exactly this, picking through the samples. So we sort them into their various phyla and preserve them, and we need to make decisions along the way. Is this one better preserved in formalin? Is this one better in alcohol? When we put specimens in formalin, they're no good for DNA after that, because the DNA breaks the formalin breaks the DNA into small bits that are hard to put back together. So if we're going to do molecular analysis afterwards, we preserve animals in ethanol, or freeze them. But formalin, which is a preservative that's been used for decades, is very good for morphology because the animals remain quite soft, and when you put them in ethanol they become brittle, and it's not so easy to study them when they've been in formalin. So ideally, if you've got lots of time, you might put some in formalin, some in ethanol, but if you don't know what species they are yet, it's a bit hit and miss, but that's just part of the process. And as Letitia mentioned, for some animals, we do live animal photographs back in the lab because, for example, nudibranchs, when they're preserved, they look like bits of chewing gum, just sort of grey colour, and the colours and the patterns are very important for identification. So we always photograph nudibranchs before they're preserved. So next slide. So this is just reiterating what I was talking about before. So what we might do when we're collecting for molecular work, we need the animal for the morphology, but we need the DNA quite often to assist with the identification, and also to look at the evolutionary relationships among the animals. So we might relax the animals first, take a small tissue sample, for example with this nudibranch, and preserve that in ethanol or freeze it at -80 so it can be sequenced, but we always need to keep a voucher specimen so we can go back to it after the DNA is done. You want to have the animal intact. So next one. So this is what we've got in our garage. Probably the owners of the property might be a little bit surprised if they walked in right now, but so we packed the specimens for transport, and we'll be going back to the museum on Saturday. Next one. And when we get back, a very important part of our work is documenting. So when we're in the field, we'll make field notes, how many of whatever we've found. When we get back to the museum, every specimen lot, which we count as one or more of a particular thing, gets a registration number. We document where it was from, when, that the habitat, in this case will be saying Tathra Wharf and which pile it's come from, and that gets put into a database. Next one. And it is then shared with other institutions and makes that information available. So people sometimes think museums are kind of dusty, crusty places. A museum is like a reference collection for animals, but unlike a reference library of books, the more we work on them, the more value they've got, because we're adding information to that specimen when we get it out of the jar and study it. And taxonomy can be a changeable feast. In the case of molluscs, a lot of them, the molluscs were first described just on the basis of the shell, but now people are more interested in the soft parts of the animal, so we're doing molecular work, and lo and behold, what was thought to be one species might turn out to be more than one on the basis of the molecular work, or it can turn out the other way too. People have separated them on the basis of differences in their colour pattern, and then we look at them and no, they're actually just one species, and instead of a whole bunch of species, we've just got one. So this is an ongoing piece of work, and as a collection manager, when the taxonomy changes, we've got to change all the labels, we've got to move them around to their correct place in the collection. So it's a process that is ongoing. They're not just, the specimens aren't just sitting on a shelf gathering dust. We're always updating the collection and updating the taxonomy, because if we have somebody wanting a specimen of a particular species, we need to make sure we've kept up with the taxonomy so it's in the right place. We know what it is, we know where to find it on the shelf, and so it's a lot of work actually. So our electronic data is made available to an international audience through the Atlas of Living Australia, and the iNaturalist data that Libby's involved with gets loaded into this as well. So people can do searches in the Atlas of Living Australia by locality, so it might say Tathra. You can pull up all the molluscs known from Tathra, and that's what we've done, and we'll be cross-referencing what we've collected against that species list, and anything that is new or we haven't got on our list could be a new record for Tathra, so that's important information as well. One reason, and I'm talking too long now, I can tell, it started and there's no stopping, but one reason we're interested in coming down here particularly is that the distributions of a lot of animals, both vertebrates and invertebrates, are extending further south, because with climate change, the East Australia current is now moving further south before it goes out towards New Zealand. So larvae and juveniles of certain animals that used to live in much warmer water are now shifting further south, and a lot of you have probably noticed this thing, animals turning up here that you haven't seen before. Whether they stay for a long time and settle depends on a number of factors. The water at a particular time might not be sufficiently warm for them to stay and reproduce, but something that with our data we can feed into databases and say, okay, 200 years ago when we first started our museum collections, that species wasn't found here, but now it's turning up here. An example of this is the common Sydney octopus, which I saw a lot of those under the wharf, and I didn't collect them because they're you know, they look at you. But I am interested in squid and cuttlefish, and octopus is my sort of specialty group. But we know they're here already, so, you know, there doesn't seem to be a lot of point making them unhappy just yet. But, yeah, so the common Sydney octopus was only found on mainland Australia, but with the East Australian current carrying the tiny little octopus larvae further and further south, they're turning up in Tasmania, and that information came to us through lobster fishermen who said, oh, we've got another octopus coming into our lobster pots and eating our lobsters because they love crustaceans, and it's a range extension of that particular octopus, so it sort of has effects all the way along the chain. So what else do we do at the museum? We do outreach and education. We talk to school groups, Science Week, public talks like this one, and it's a great place to work. So it's not a feisty, dusty old, you know, collection of jars. We're all busy doing things and recording Australia's biodiversity, and that sort of information is going to be available for centuries to come. So that's my spruik for the museum.